light on data, light on results, and heavy on, thank you, kind of big ideas presentation. So it's the perfect kind of after lunch edutainment. And what I decided to talk about today, in keeping with the theme of the conference, is what I think we need to do to get to a place of what myself and some colleagues are calling digital pathogen surveillance. And you'll find out exactly what I mean by that over the course of the presentation. But the perspective that I'm coming from is as somebody who sequences loads and loads of pathogen genomes. And I've seen genomics, first of all, become a really useful tool for outbreak reconstruction, and then genomics become a really useful tool for sort of epidemic and pandemic level dynamic analysis. And now the idea with digital pathogen surveillance is, can we take genomics and turn it into more of a predictive tool? Instead of responding and reacting to outbreaks and epidemics, can we be a little bit more proactive about preventing them in the first place, or keeping outbreaks from turning into epidemics? And if we're going to do that as a genomic epidemiology community, the sandbox that I play in, we need to start making friends in other sandboxes, and I really hope that's something that comes out of this meeting. And there's a couple challenges that we need to address, not just sort of methodological and research challenges, but really big picture things. So that's what I'm going to talk about today. A lot of the thinking that I will dispense upon you in the next 40 minutes or so actually comes out of an article that's coming out soon-ish in Nature Reviews Genetics. They commissioned Nick Lohman and myself to write on this digital pathogen surveillance and our perspectives on what's going on. So there's Nick, there's me, there's our Twitter handles. There's bonus Bill Hannage for the evolutionary biologist folks in the room. We always joke that if we started a band, this would be our band cover photo, our publicity photo. We'd also not be very good, probably. But yeah, a lot of this thinking is really me and Nick bouncing ideas off each other for the last little while. And the foundations of the Nature Reviews Genetics review that will be coming out soon were actually laid in an article that Nick and Andrew Rambeau and I did, just a little short perspectives piece back in 2015 in Genome Biology that really got us thinking about this in kind of a bigger way. So this all comes from that sort of brainstorming process. I want to start with a little trip into the literature and a specific slice of the literature that I like to call the pandemic lessons learned literature. In the wake of any major outbreak, epidemic, or pandemic, there's always a bunch of these lessons learned papers that show up. People saying, oh, you know, what can we do better next time? And this is not a new thing. The very first one I was actually able to dig up comes from 1872. It was a bit more clinical. It was on lessons learned from a smallpox epidemic. But the lessons learned in this sort of modern communicable disease era, I guess the first paper that really popped up was this one, Science 1919. This is Major George A. Soper, who was a sanitation engineer. He was famous for discovering who Typhoid Mary was in the States. But he, in the wake of the 1918 influenza pandemic, wrote this piece on lessons learned. And the lessons at the time, back in Major Soper's days, were really kind of around understanding the pathogen itself and understanding how we could protect ourselves. This really reflects kind of a lack of general microbiological, virological knowledge of the time. If you fast forward to the next lesson learned paper in our little time machine, this one is from Mac Shanholz, which is a fantastic name. And Mac here is again writing on influenza. This is the H2N2 outbreak of 1957. And here the lessons switch more to what can the health system do to prepare for future pandemics. This, I think, really kind of sets the stage for the current genre of pandemic lessons learned literature. And he says things like, we've seen the importance of cooperation between state and local health departments. Future epidemics could be handled easily if communities were organized. And this paper also marks the beginning of what I call the pandemic lessons overconfidence era, One of the first sentences in the article is, "We learned a great deal from the experience of planning for the present pandemic." And obviously they didn't, because you know what kept happening? More pandemics. We fast forward to 2003 and SARS, and we get more of this pandemic overconfidence. By providing a test of the capacity of the public health system, SARS has better prepared the world for the next pandemic. Spoiler alert, it didn't. H1N1 showed up in 2009, and you saw the same thing in the post-H1N1 pandemic lessons learned literature. Oh, this has made us better prepared for the next pandemic. And it doesn't. We think if we just articulate the lessons learned, that somehow we will be able to internalize them and act upon them. And this isn't true. This is Bill Nye the science guy. I do a lot of science communication, and one of the things that we teach in science communication is something that is wrong, something called the knowledge deficit model. The knowledge deficit model is what you don't want to do in science communication. It basically says people just need the right information, and they will be able to take that information and act upon it accordingly and make the right decisions. So what we're doing with this pandemic lessons learned literature is essentially the sort of knowledge deficit model. If we just somehow articulate the lessons that we learned in this pandemic, this epidemic, surely it won't happen again. Surely somebody will listen. And in science communication and risk communication, we'll tell you time and time again that the knowledge deficit model is false. You can't just give people information and expect them to act on it properly. So why do we as a community think that if we just say, oh, well, these are the lessons we learned from this pandemic, people will act on it and we'll get it right next time? We don't. Ebola happened. Ebola first popped up in December 2013. It was undiagnosed for months. Even once it was diagnosed as Ebola, there was several more months of people just not paying attention. Zika, same thing. It was circulating undetected in Brazil for ages before a bunch of docs who were using WhatsApp are like, hey, something interesting is going on here. Clearly, what we're doing in terms of pandemic preparedness isn't working. And if you now look in the pandemic lessons learned literature in the wake of Ebola, in the wake of Zika, that pandemic overconfidence that, we're now better prepared for the next one, that's essentially disappeared. And finally, it's been replaced by the truth, which is that we're really not very well prepared to handle the next big outbreak, the next big epidemic, and the next big pandemic. We need to do something different. What could those different things be? Well, if you look in that lessons learned literature, there's a lot of good stuff, things around governance, things around enhancing local laboratory capacity, things around building up better communications infrastructures. But one tiny little slice of what I think could be a sort of a new approach to potentially preventing outbreaks from becoming epidemics, from becoming pandemics, is this digital pathogen surveillance. So when I talk about this, sort of as articulated by Nick and Andrew and myself, it kind of exists at this union of the Venn diagram of three areas. And we can probably add more into this. I'm sure at the end of this meeting I will be inspired to have added another fourth circle or something. But we've got genomic epidemiology, which I'll kind of broadly define as the use of genome sequencing data to understand outbreaks and understand epidemics. Everything from individual person-to-person transmission events to helping to calibrate some of the models we use to understand epidemic dynamics. Then you've got digital epidemiology, tapping into really unique data streams to get a better sense of what's happening in a population. And then you've got the One Health ethos, which maybe people might not be as familiar with. And this is the notion that animal, environmental, and human health are all so intimately interconnected that disruption in one bit of that landscape will cause a ripple into the other domains, and that if we want to be at all proactive about identifying spillover events, emerging infectious diseases, we really need enhanced surveillance that doesn't just look at humans, but also incorporates veterinary surveillance and incorporates environmental work. So I think that if you sort of take things from these three areas in a way that I will attempt to articulate over the rest of the presentation, I think you end up with digital pathogen surveillance and potentially a new approach to responding to outbreaks and hopefully making sure that outbreaks don't turn into epidemics or pandemics. So I'll go through each of the areas in turn. I'll spend the most time here because this is what I know. They always say talk about what you know, so this is what I'll talk about, genomic epidemiology, but I'll mention a bit about how the digital epi and the One Health stuff will fit in. But let's start here. When we talk about genomic epidemiology and the use of genetic sequence data to help understand outbreaks, to help understand transmission, there's quite a long history, and we heard some of that earlier, looking at polio gene sequences, certainly looking at viral gene sequences in HIV, and Hep C has been done for many years to really understand transmission. But this field really kind of blossomed just in the last maybe 5 to 7 years. Our group was one of the first to publish complete outbreak reconstruction using genomic data, and this was a tuberculosis outbreak, a quite large one that had happened in BC. And at the time, and for the first few years after this came out in 2011, these sorts of analyses were generally done retrospectively. Oh, an outbreak happened a while ago. We've got the isolates in our freezer. Let's take a look and see if we can draw any interesting conclusions that are now, far too late to save the day, but we get a nice paper out of it. And we also, at the time, these first sort of early outbreak reconstructions that we did were very, what I would describe as kind of methodologically back of the napkin calculations. There was nothing really formal about the way we were drawing inferences around transmission. Same with the other groups that were doing this. It was really just kind of identify SNPs and, draw some arrows between people on your phylogenetic tree. We realized that was an entirely unsustainable model, so we moved towards more modeling-based methods for essentially kind of automated reconstruction or automated inference of transmission from genomic data. Our first kick at the can was some years ago, and we've improved on that significantly since then. This was out in MBE earlier this year, and this is all work with Xavier and Caroline at Imperial. So this really took our genomic epidemiology into a space where it didn't require so much manual intervention. We could instead feed genomic data from larger outbreaks and larger sets of outbreaks into these methods to kind of give you a bit of an analytical kickstart. We also, in that same transmission inference work, have realized that you can, in addition to sort of inferring putative transmission events and their direction, you can also infer the timing that those transmission events happened on. So this was, I guess, last year in microbial genomics. We used a variation on our transphylum method to, in a large outbreak of tuberculosis, a different large outbreak. We have a lot of large outbreaks of TB in BC. This one, it was in this lovely waterfront town here in the background. But we had a very big outbreak, and with this method, we were able to determine when people were infected with TB and when it actually sort of woke up and they were diagnosed with active disease. There's latency in TB, so some people get infected and progress to disease right away. Other people have this fairly long, drawn-out period of maybe a year or two, and that's really useful information if you're trying to understand what's happening in your outbreak. Are the cases you're seeing towards the tail end, are they the result of people that have just recently acquired their disease, transmission is still happening, and you need to keep your public health response going, or were they infected further back in the outbreak and are just kind of waking up now? So we've developed methods to do interesting things, like infer transmission, like infer timing of transmission, and we're now doing things on much bigger scales too. One of my PhD students, we've gone through, opened up the freezers at BC CDC, dumped out all the TB that we could find, and looked for every case of TB that shared a DNA fingerprint with at least one other isolate. That was around 1,200, and we sequenced all of those and some extra ones just for fun because we had the money to do it. And so we've now built a complete phylogeny of every single case of clustered transmitted TB in BC going back a decade. So we've really, in the past 5-6 years, has gone from sequencing one outbreak very back of the napkin to just, sequencing all the things. And what's cool too is that this work, although to date it's been very retrospective, it's now becoming prospective, and we're starting to sequence all the TB that comes in our door so we can identify clusters in real time. This kind of real-time genomic epidemiology era was really, I think, birthed with a couple papers. One that Nick worked on was a very cool example of a hospital outbreak of salmonella in the UK, where with nanopore sequencing, within about an hour of getting DNA out of a sample, the team was able to either rule in or rule out that isolate as belonging to an outbreak in real time in the hospital lab. The realist time deployment of genomic epidemiology, though, certainly was with the 2014 Ebola outbreak. Here's Raymond working in the lab with the MinION, actually doing sequencing right here. There's Ebola virus sample on this. And this is, I think, a watershed moment in the genomic epidemiology field for a couple of reasons. Reason number one was you really saw the individual kind of outbreak level reconstructions. This person transmitted to this person. We think this person transmitted to this other individual. These cases are linked. These cases aren't linked. That individual genomic information was being fed to epidemiologists in real time who were using that data to manage their contact investigations. Nick would CC me on lots of the emails that were going around where they were sharing the genomic data and the phylogenomic data with the epidemiologists and answering their questions, and it was really interesting to see this play out in real time. And I think the second reason that this was really kind of a big coming out party for genomic epidemiology was that it scaled up from not just that sort of person-to-person transmission reconstruction that we've been doing for ages, but really into this full-scale phylogenetic type of reconstruction, exactly what Gita's presented this morning, where on country-wide levels, you can now start to make inferences about what's happening. You can really understand the epidemic dynamics, get information that feeds back into your epidemiological model. So it was really genomic epidemiology on both small scales and very big scales, which was incredibly exciting. Now, when we're sequencing for outbreak reconstruction, inferring epidemic dynamics, all of that jazz, oftentimes we are sequencing from pure culture, and that takes a while. You might be sequencing from amplicons, PCR amplicons of a virus that you're after. You might be sequencing from, TB that you've scraped off a culture plate or some liquid culture medium. But what if instead of, waiting for pure culture material, which can be days, weeks in the case of TB, even months sometimes, what if we could turn our sequencing results around in the space of hours or a day as opposed to, weeks or months? And what if we could couple that genomic epidemiology, sequencing to uncover all these interesting insights, to the actual diagnosis itself? So that's what we call clinical metagenomics, and the notion is that you take a sample and you sequence directly from that sample all of the nucleic acid that happens to be present, and you say what's in here. This is an example of one of the tools that's out there to do that bioinformatic assignment. It's a taxonomer from Robert Schleberg's group. And this is a sample where you can see there's, some human reads, some bacterial, lots of viral reads. If you were to click on the virus purple button, this little purple circular diagram pops up, and it shows you the various taxonomy of that virus. And you see that the majority of the viruses is Zaire Ebola virus. So you were able to make a diagnosis of Ebola virus directly from a blood sample and recover a genome, which you can then use for epidemiological analysis. This is why I'm better in science than I am in art. It's probably a good reason or a good job I didn't actually go into any sort of creative artistic field, because I'm not really good at it. Nature asked us to provide some figures for the review, and they said, we have our own art department, so you don't have to worry about making a nice figure. Just kind of tell us roughly what it should look like, mock it up in PowerPoint or something, and we'll make it for you. And I had this vision of let's draw a mobile medical unit, and I don't really know how to draw a mobile medical unit in PowerPoint, so I just sat at my desk with a pen and came out with this. And this stinker of a diagram is kind of meant to represent the challenges that clinical metagenomics faces. What are some of the technical hurdles that we, as a sequencing community, are going to have to get over if we're going to use genomics in real time? And those challenges exist at various points. You've got, your physician treating a suitably sick and confused-looking patient here. You've got a technician processing some samples. Your bioinformatician, she's running, this is not a tampon. This is supposed to be the MinION sequencer plugged into what is clearly an Apple laptop. And then you've got either your clinician or your epidemiologist here who is holding a report in their hands, and everybody's got round feet for some reason in my world. Some of the challenges at each of these levels, if we look at pathogen, genome size, the smaller the genome size, the less likely it is you're going to be able to capture it in your clinical metagenomics result, especially if you've got a low abundance sample. The host, phase of infection, viral titers will vary over the course of an infection. In TB, the amount of bacillus that you cough out on any given day, varies. It's higher in the morning, it's lower later in the day. Abundance of the pathogen within the sample that you've got. How stable is that sample? Is there other DNA? This is why sequencing from sputum in TB is really difficult. There's all sorts of other stuff kind of drowning out the TB signal. In terms of getting the sample ready to go, do you need to do any enrichment steps? Library prep is a giant pain in the butt. We know that reagents are contaminated all the time. Sequencing, how expensive is it to sequence? Is there any real sort of cost-benefit driver for being able to do this? How portable is the sequencer? This is an extremely portable sequencer, but if you've seen Ian Goodfellow talk about his work sequencing in the Ebola outbreak, he went with the ion torrent sequencer because it was the one that had the fewest moving parts inside. So if it broke down in the field, he and his team would be able to fix it themselves, as opposed to having to call in a tech to West Africa to fix a broken sequencer. So how portable is it? How good is the sequencing technology? In terms of the bioinformatics, there's all sorts of challenges there. Do you have the computing power that you require to run these sorts of clinical metagenomics bioinformatics pipelines? Do you even have power? Can you actually power your computers? Can you connect to the internet if you need to do any of this in the cloud? And then some other stuff like, how complete are the reference databases that you're using? If the sequence isn't in there, you're not going to find it. Is your diagnosis going to be complicated by close relatives that are in the databases? How good are your bioinformatics pipelines? And at the end, are you able to, with all this genomic data, are you able to generate some sort of interpretable output, no matter who your end user is, whether it's the clinician, whether it's the epidemiologist working in the field, and do they see from that report what the action that they should take is? And in terms of these challenges, there's not a lot that we can do about the first two there. And lots of people are kind of focusing all sorts of interesting research programs on, all of this bit in the middle. One of the bits that I personally think has been kind of ignored and underrepresented is this latter part. How do we take this genomic information that we're generating and communicate it in a way that is both useful and usable to our end users? Public health on the ground, it's not people like us. It's epidemiologists, it's clinicians, it's nurses, it's public health officials, it's NGO workers, and many of them have never seen a genome before. So how do you make that information interpretable and actionable? Tiny detour on that topic. This is some work that we're doing right now in collaboration with Public Health England. They've now rolled out routine genome sequencing for TB diagnosis and sort of predictive typing. They wait until a sample turns culture positive in liquid culture, extract the DNA, batch it, run it, analysis proceeds on a central server, and a report is generated automatically at the end, and that report goes to the various end users, some of whom are clinicians, some of whom are surveillance people, few of whom are people trained in genomics and bioinformatics. And this is what the report looks like now. I won't walk you through the whole thing, but suffice to say that even for the experts in the room who are very comfortable with TB genomics, some of the stuff is a bit tricky to parse. Some of the way they report out resistance typing, these little codes, what does S stand for? For somebody that's not familiar with genomics, all of this, organism identification, the first thing you see under there is Kraken. It's like, oh great, I've diagnosed my person as having a sea monster. What does it mean with human? The percentage of human, what's good, what's bad? This is a very difficult report to interpret. So our group, in the interest of showing the community that thinking about how we report information is sometimes just as important as the information itself, embarked on what's called a design study. This is methodology borrowed from the field of information visualization. One of my grad students, Anna Kersan, she's co-supervised with an InfoVis researcher in computer science at UBC. And so InfoVis, in a design study, your goal is basically talk to your end users, see what their tasks, their workflows are like, understand what type of data they work with, come up with a series of visualizations or visual representations of that data, trial those out with your end users, see what they like, see what they don't like, and use that to arrive at a final visualization system, which in this case is sort of a final report for this genomic data. And it's something that I think is anathema to many people in the genomics and bioinformatics and math community. It's mixed methods research. We had to put our social scientist hats on, and it got a little weird, and there were a lot of words that we didn't know. But we've taught ourselves things like the exploratory sequential model and the embedded model and how to do semi-structured interviews and all that good stuff. And it was really interesting. We developed some surveys, and we did loads of stuff in this. The surveys ask things like, okay, if we give you 4 different representations of an element of the report, and in this case we're trying to see what is the best way to draw attention to which drugs is this isolate resistant to. Do we just let the user get that information for themselves? Do we shade it? Do we have a little alert glyph? Do we bold it? And then we ask people, hey, rank your preferences. And when we do that across loads and loads and loads of different choices, whether it's the way we word things, the way we visualize or present them, we end up learning a whole bunch of really interesting things. The first thing that I think is really important is that when you go out and talk to your end users about how you're communicating genomic data to them, you always learn very, very surprising things about their perspectives on that data and how they're using that data and some of the constraints that surround how they use that data. Nick, for example, when the Ebola work was going on and he was sharing some of these email chains back and forth, he was always really surprised that stuff that seemed very obvious to us as trained or phylogeneticists was being interpreted completely differently by epidemiologists. We think of a phylogenetic tree as something that would be fairly straightforward to read, but they were reading entirely different interpretations of that tree. So it's very important for us as, domain experts in one area to understand our users' perspectives. We also learned all sorts of stuff that sort of constrained how we presented the report. And the other important thing is that we compared our designs to what was on the existing report. So we call that sort of the control report. And in virtually every single instance, the designed design, the one that we came up with after user consultation, always outranked, always outperformed the control design. So just by virtue of stopping to talk to our end users and get a sense for what they wanted from the genomic data, we are able to design a report that was much more palatable to them. So, in terms of genomic epidemiology, how it fits into this notion of digital pathogen surveillance, it's the part of that Venn diagram that can accelerate diagnosis. If we can do clinical metagenomics and turn results around in hours or days, we can tell people what they have, identify what an outbreak is as it's happening, and by getting genomes out of that diagnostic procedure and either looking at individual person-to-person transmission or using a more phylogenetic approach to understand dynamics overall, what can we learn about that budding outbreak that might help us control it before it gets any bigger? So that's piece number 1. Piece number 2 is where digital epidemiology comes in, because it's great, with genomics, we get called by the WHO, for example, to say, hey, we've got a weird outbreak of something, can you come do this? Or I get a call at my desk, we've got some weird TB happening in this community, can you come check it out? But that's really still being reactive. How do we get this to be more proactive? And there, I think there's a case to be made for embedding this sort of genomic epidemiology into routine surveillance efforts. So that's where digital epidemiology comes in, and the notion that there's all these interesting, cool data streams out there that provide a different look at disease dynamics than traditional surveillance streams do. What if we can tap into those and use it to identify outbreaks right when they're starting, instead of waiting for a call once the outbreak spiraled out of control? So here's an adorable cat working at the computer. If you are lucky and you work in a fairly well-resourced setting, like I do in British Columbia, we've got traditional surveillance systems that are looking at things like lab test positives, medical services plan billings, just calls coming in from clinicians on the front line. We've got a team of surveillance analysts who are actual humans, they're not cats, but they're sitting there at their computers on a weekly basis looking for interesting things. And if something alerts, something's happening at a level that is higher than we would expect it to, that sort of triggers follow-up kind of up the epidemiological food chain. But this sort of surveillance system, this notion that you can detect, you can report, and you can act, doesn't exist in most settings, particularly settings where outbreaks are likely to become epidemics. There's no traditional surveillance system. So how then do you capture information that says, hey, a disease event is potentially happening here? That is where digital epidemiology comes in. I am a huge fan of digital epidemiology. I don't participate in it. I'm just kind of like the enthusiastic observer. Marcel Salate will talk later in the conference, and he's the expert, and he will tell you all this amazing stuff. I just kind of stand on the sidelines and go, oh, this stuff is really cool. But I'm hoping to get more into it. One of the reasons I'm really excited about being at this meeting. Digital disease detection, digital epidemiology, I'm sure you're all familiar with the basic premises of it. It can be really sort of gonzo, it can be really traditional and cool, but it really kind of leverages the fact that media and social media exist in places where public health infrastructure doesn't. And if we can tap into these digital streams, whatever they are, we can get a better picture of disease events. Those might be really kind of crazy digital streams. This is Adam Sadilek's Nemesis project, looking at Twitter for signs of foodborne illness. Super cool. Or, like I said, they can be more sort of traditional digital surveillance, but still incredibly powerful. Healthmap.org is the classic example from John Brownstein and his team, a fellow Canadian. Here's what's happening in and around Hong Kong this week. Really amazing stuff. And just thinking about HealthMap and how it unfolded in Ebola, I think really gives us a lot of hope for this as a tool that can give us that kind of early alert system. So if you look, the very first sort of health map inkling that something was happening appeared on the site on March 14th. There was more news on March 19th, more news again on the 21st, and March 22nd was when the outbreak was actually recognized as Ebola, and it started to appear on other people's radar, and you'll notice a fairly steady stream of alerts after that. But HealthMap had March 14th to the 22nd, this is like an 8-day head start. You've got a little window here. What if we could have deployed sequencing there? What if we could have got a diagnosis earlier instead of waiting until the 22nd? What if we could have even deployed genomics at any point along here? And it wasn't, you'd have to follow that line way out down the road to reach the point when it was actually deployed. So digital epidemiology can kind of give us this early alert. Hey, something's happening, maybe we can be a little more proactive about getting sequencing-based diagnostics and genomic epidemiology into a setting where an event is unfolding now. Now, where does One Health fit in? Well, One Health, I think, is maybe the idea that's going to help take this whole thing kind of to the next level and help us get to where the next outbreak is likely to happen before it even starts, before it becomes a blip on the health map radar. So as I said earlier, One Health is really about enhanced surveillance that combines animal, environmental, and human health together into one package, kind of looking for potential spillover events. And it gives you that really kind of early warning system. This is a photo I took years ago. We were doing some CBC television stuff in a town in Mexico, and this is sort of an example of the types of places where One Health works. When you've got, you can't really see them in this picture, but this is sort of the animal barn. There were some pigs, there were some horses, there were some chickens. Everybody occasionally spent time in the barn, but the animals would wander all around in the yard, and the people's house was about sort of, maybe 3 meters in this direction. So the health of all of those groups were quite intimately linked. Diseases, when they show up for either the first time emerging infectious disease or when they start to turn into an outbreak, maybe they've entered a new geographic area, they don't come with birth announcements. It would be great if they did, like, hey, congratulations, Brazil, you've got Zika now, but they don't. We have to rely on surveillance systems to identify these things. And with digital epidemiology, those surveillance systems can be pretty awesome, but it would be great to know really where we should be looking for these events before they happen, so we never have to get this birth announcement. And we can do that. We have maps that tell us where to look. This is work from Kate Jones, who's part of Peter Daszak's group, the Eco Health Alliance, who does so much amazing stuff in One Health. This is from 2008, and this is essentially a disease hotspot map. Where do a variety of factors converge? Factors that, when combined with each other, lead to a higher risk of spillover events. And these are factors like wildlife, biodiversity, demographic shifts, people increasingly adopting farming and agriculture lifestyles, civil unrest, and very obvious things like breakdown in local public health systems. All of these things point to where new infectious diseases are likely to emerge from, where new outbreaks are likely to happen. And even though this work was 2008, it was actually pretty prescient around getting the emergence of H1N1 correct, and even West Africa as a hotspot with the 2013, 14, 15 Ebola outbreak. So this map seems to work. This is a nice paper from Simon Anthony, who's part of that whole Eco Health Alliance group, that by looking at feces from macaques in Bangladesh from a variety of different communities, they were able to establish that viral diversity and viral communities are somewhat, at least, deterministic. They're not entirely stochastic. There are patterns there. And if there are patterns, that means that we could potentially predict, identify disruptions in those patterns and use it as sort of an early warning signal of a spillover event. Just another cool little knowledge translation thing. Simon realized that the content of this paper was quite dense, and many of you have probably read this, and it's kind of a tricky one to get through. It combines modeling and ecology and sequencing and all sorts of good stuff, and he was having a difficult time communicating it to funders. So he approached a friend of mine who runs a data visualization studio and said, hey, can you come up with a way that we can make this paper and its findings make sense to a broader community, like the type of people that might be funding pandemic preparedness responses, philanthropic foundations, for example, just end users in the community. And so my super good friend Jer Thorpe has got this data viz studio, and he put together this cool website, let'spredictviruses.com, where it kind of explains Simon's work and even has a little game built in. There's some little poop emojis. It's kind of a neat tool for explaining a very complicated idea in a way that makes delightful sense and is accessible to a bunch of people. So, thinking about the role of One Health in this digital pathogen surveillance culture, it's really kind of acting as our distant early warning system. So if you put the three things together, you end up with a model that looks like this terrible drawing, also from my own hand. This is going to be figure 4 in the review article, but this kind of sets out this future digital pathogen surveillance model. You've got your hotspot maps that tell us where do we need to be doing enhanced kind of One Health style surveillance, and you've got a team on the ground that's doing, regular kind of, let's call it low-level surveillance. They're out there, they're sampling wildlife. These are birds or bats. You can read into them what you want. They're sampling domesticated animals, leggy chickens, and this very round pig. They're sampling humans that are at the human-animal interface, farmers, veterinarians, for example. And they're just looking to see, what's there. Are there any signals that might indicate spillover? If you're doing genomic analysis, maybe you're using clinical metagenomics to identify what is in the sample that you take from your pig here. You get a couple of viruses, you assemble them, you can do some phylogenetics on that, you can look at effective population sizes, you can say, is something growing? Are we seeing more of this? Maybe you start to see spillover events, farmer gets sick, visits a doctor, they enter some data into a billing system, maybe the media. Locally, there's a reporter with a little press thing in his hat and a little notepad, obviously. The reporter catches wind of, there's a bunch of cases of something going on in this community. That triggers alerts on Health Map. All of that combines. The digital epidemiology sends signals that, hey, we've got an actual kind of emerging event. That goes back to the team on the ground. They scale up their genomic response for diagnosis and epidemiology. Sequences are constantly being posted online. They're being shared with the community, analyzed in real time. That information is being disseminated to everybody who's interested, and hopefully you can attenuate an outbreak before it gets too big. So we'll finish with a couple of the big picture roadblocks. I've shown you, there's roadblocks related to all the different aspects of clinical metagenomics. There's roadblocks related to how we communicate these results to stakeholders, but there's other ones too. And I'll highlight 3. So the first is kind of captured by this tug-of-war diagram and the notion that there's this tension between the way we're kind of responding to outbreaks in this research-based fashion, which is what we're doing now, and actual clinical, real public health use. This encapsulates a lot of things. First is that if you want to take a very researchy tool like genomics and put it into the clinical space, all your stakeholder audiences have various evidentiary thresholds that they need to have met if they're going to say, okay, I accept this technology and I will use this routinely. And we don't have any idea who really all our stakeholders in genomic epidemiology are, let alone what their various evidentiary thresholds for accepting this technology are. And we cannot do what we're doing now, which is have our outbreak responses driven by a bunch of boffins. It's very exciting for us to be part of an outbreak response, but, most of us aren't public health. I work at CDC, so I am half public health, half academic boffin, but we can't have outbreak responses driven by research teams and PhD students and postdocs. It's a non-sustainable system. We have to avoid what I call genomic paternalism, the notion that, these smart researchers from around the world will sort of fly in and solve your problems and then disappear, leaving you no better than what you started with. And this isn't always the case, but we have to be cognizant of this as a trap that we can easily fall into. And we also, we get super enthusiastic about the work that we do. And we're like, this stuff is great. It can save the world. And so we have to balance our enthusiasm for the work that we do with kind of overselling its utility. And I have to say, I've seen a number of clinical metagenomics applications. We see the good stories get published, but for every good story, there's 10 or 20 stories where you didn't get an answer, or you got what you thought was the answer, and then it was wrong, and it turned out to be something else you thought, and then that turned out to be wrong, and then you go down this laundry list of pathogens. So there's challenges here. There's challenges in the ethical domain. The ethics of public health is super different from traditional medical ethics. There's all sorts of issues that we need to deal with here. What if we find incidental findings in clinical metagenomics? What if you're trying to diagnose somebody with flu and reads from HIV or Hep C pop up? Who owns pathogen genomes? Who owns metadata, or just data, as we've been calling it today? Nobody knows. And now, with our ability to infer transmission, direction, timing for bacterial infections, the HIV and the Hep C community have been dealing with this question for a while, but now this is unfolding on a broader scale. What are the ethical implications? What are the legal implications? And finally, data sharing. I'm not even going to go into all the problems with data sharing. It has been written on many, many, many times before. It's been mentioned many times today. If we don't share, we don't get anywhere. The Care Bears want you to share. Sharing is caring, and we need to keep that near and dear to our hearts. This is a really good example of that. Here's an article. I've blacked out the authors' names to prevent them from being embarrassed. Sharing data. Barriers for global sharing exist. No shit. It's behind a paywall. As Christian Anderson pointed out, they are doing it all wrong. So we really, as a community, need to think about how we're sharing our data. Excellent examples come from Andy's virological.org, Trevor and Richard's NextFlu. Watch out very soon. Next Mumps will be released. Ourselves and the Broad and Trevor's group have contributed a load of mumps genomes that we've been sequencing from the ongoing outbreaks in North America. So you'll see a new next month very soon. So to conclude, what do I think we need to do to get to digital pathogen surveillance? Each of us in those individual circles on the Venn diagram need to think about some of those big picture issues that I mentioned, not just the technical challenges. We need to be concerned not with our own research programs and developing cool new methods and doing cool new analyses, but also start to think about those bigger picture things like the ethics, like those tensions, like the data sharing. And really need to cross-pollinate. We need the digital epi people talking to the genomic epi people, talking to the One Health people. If we do that, we might exist in this very exciting and very poorly drawn future state that I think will be a really interesting place. So there's more cats. Thanks to lots of people. And thank you all for listening and staying awake after lunch.